This is the basic genetics lecture. We'll be going over the study of heredity, or how traits can be passed from parent to offspring. A lot of this is review if you've taken any type of life science class previously. So you may be familiar with some of these topics, and a lot of this could be a review for you. So let's go ahead and get started here. First, we want to discuss how traits are determined, and scientists used to think that it was a blending of traits, so parents were blended or mixed in the offspring. And we do see a lot of things that resemble that now, but there's not just a pure blending between the two parents to create the offspring. Otherwise, all the offspring would look very similar. Sometimes siblings do look very similar, but a lot of times siblings do not. It is not just a straight blend like this would be. It's not just going to take two people and blend them together to make a child. The offspring has their own unique characteristics, not just the same characteristics from the parents blended together. So the person that first started to focus on this was Gregor Mendel. He used pea plants to study how traits were passed on. Why did he choose a pea plant? There were three specific reasons. First, the pea plant self-fertilized, so a pea plant can be both the mother and the father to the peas. That allowed Gregor Mendel to know exactly who the parents were and allow traits to be passed down very quickly. Two, they have distinct traits, which means that each trait is either one way or another way. There is no in between. So either the plant is tall or it is short. There is no medium size. The pea plants either have white flowers or purple flowers. There is no light purple. They either have round seeds or wrinkled seeds. There's no in between. So the traits are very distinct. They also have a rapid reproduction cycle, which means that he can go through many generations in a very short period of time. So he used over 20,000 pea plants to gather information and then used statistics, which is math. He disproved that the blending hypothesis was occurring, and he came up with his own method that we now call Mendelian genetics. So organisms that receive the same genetic traits from both parents are called purebred. We also use purebred to refer to types of animals that are in a direct lineage, such as, purebred dogs or purebred horses. Purebred in this case, in this class, is going to just mean that they have the two of the same traits. And hybrid is an organism that gets different genetic forms from each parent. So you might have heard things like hybrid cars, where they use some gas and some electricity, or a blend. Other organisms, we'll use hybrid later on in this semester to describe some organisms as well. But in this case, for genetics, a hybrid just means that they have one trait from one parent and a different trait from the other parent. So when we cross these traits, Mendel went through three generations. He started with the parental generation, which was pure breeding plants of two different traits. So then he crossed those two plants and got the first filial generation, which is the first offspring. We often refer to that as F1. Then allowed the F1 to self-fertilize, which gave us the F2 generation, or the second filial generation. He did not see any blending in these pea plants. What he saw were very distinct traits. If he took a pure breeding yellow crossed with a pure breeding green pea plant, all of the offspring were yellow. And then if he allowed those yellow to self-fertilize, he saw the re-emergence of the green trait. He did also see that he did look at several different traits as well. If you look up at the top picture up there, he looked at whether the seeds were round or wrinkled, yellow or green, the flower color, the pea pod shape, the pea pod color, where the flowers were located on the plant, and also the height of the plant. So he did these things with all of these different traits to get the same results. So what is a ratio and what is a percent? We were going to be looking at the predicted offspring from a particular cross. So if we take two parents which are heterozygous, or two parents that are hybrid, one dominant trait and one recessive trait, one dominant trait here and one recessive trait here, and we can cross those in a Punnett square to show what the predictions would be here. Okay, so we're going to be talking about both ratios and percentages to see how to determine those, and we'll get to that in a moment. The ratio here, we're looking at 3 yellow peas to 1 green. So a ratio is just comparing the numbers of different types of offspring there. And then a percentage is 75% yellow, and 25% green. So Mendel's conclusions were that traits are controlled by some factor, and that factor happened to be genes. He came up with 3 laws that control how genes are passed down. The first one is the law of segregation. And the law of segregation states that organisms inherit 2 copies of each gene, one from mom and one from dad, and those copies segregate during the production of sex cells, or during meiosis. So each parent has 2 genes. Those segregate during meiosis, so each sex cell has only 1 copy of the gene, and then those copies combine together during sexual reproduction to get a unique offspring. So each parent gives 1 allele for every gene. What's an allele, you ask? Well, it's a distinct version of a gene. So an allele is either going to be dominant or recessive. A dominant allele is the one that is going to show, and a recessive allele is going to be hidden unless it's by itself. So if you look at this picture here with the flowers, we have the big R, that represents the dominant allele, and little r represents the recessive. If we have 1 of each, then only the dominant color shows. Notice that if we have 1 of each, we don't get a light purple, we just get the same purple because that one is dominant and it overshadows the recessive. The only time a recessive is going to show is if both alleles are recessive. This is called the law of dominance. That means that for any given trait, 1 allele is dominant and the other is recessive, and the dominant will show and the recessive will be hidden if they are both combined. We don't get a blend. So the dominant alleles we always represent with uppercase letters, and recessive alleles are represented with lowercase letters. It will be the same letter, just uppercase for dominant and lowercase for recessive. So if we're talking about pea color or flower color here, we're not going to use P for purple and W for white. We're using R's here to represent the color of the flowers, capital letter for purple, lowercase letter for white. The third law is the law of independent assortment, and that just means that all chromosomes are mixed up independently during meiosis, that they don't get stuck together, and we get all possible combinations formed in our gametes. So there's a couple possibilities here for each stage of meiosis, and notice that we get all possible combinations in our offspring, not just one particular combination. If we take a closer look here, these don't necessarily have to line up in the same order every time. So we get a wide variety of gametes, which means we get a wide variety of offspring, and that's why siblings don't all look exactly alike. So far we've been using the terms purebred and hybrid for these. We're going to switch now to homozygous and heterozygous, which really mean the same thing, they're just a little bit bigger words. Homozygous means that the 2 alleles are the same, and heterozygous means that there's 2 alleles that are different. So here we have 2 chromosomes, and then the alleles are highlighted in either yellow or blue. Which one of those is homozygous and which is heterozygous? Take a moment to think about that. Which one's purebred, which one's hybrid? This one here is going to be purebred or homozygous because both of them are the same. This one is heterozygous, one wrinkled and one round, so these 2 are different. There's 2 of them because one comes from the father and one comes from the mother during sexual reproduction, and so each organism has 2 of the alleles for each trait. We call this a genotype. The wrinkled, round, or wrinkled and wrinkled traits here. The phenotype is what the organism looks like. So genotype is its actual genes. Those are expressed by writing letters. The phenotype is what it looks like or how those genes are expressed. And these 2 words are going to be very important for you to know throughout the unit, so make sure that you understand what these words mean. Genotype, phenotype, heterozygous, and homozygous are really, really important for you to understand what they mean. You're not going to be able to do Punnett squares unless you understand those words, so make sure that you know those words in particular. Genotype refers to the organism's genes. We write those as letters. And phenotype refers to the organism's physical appearance, or how those genes make the organism look. So if we look at this, if we have big P represents purple flower and little p represents the white flower, is this genotype heterozygous or homozygous? what will be the phenotype of that organism that has that genotype? Well, since we have two different ones, that's going to be a heterozygous genotype. And then because we have a dominant allele, that is the color of the plant. So because there's at least one big P showing, that flower would be purple. Then we can use Punnett squares to determine what the possible offspring would be for any given cross. So if we have two organisms here that are both heterozygous, we put one parent on the top and one parent on the side, and then combine those alleles in these boxes here, and these represent the possibilities for the offspring. They're not always going to be in this exact proportion, but these are the possibilities here. So one parent goes on the top, one parent goes on the side. These are the gametes that the parents produce. That's why we have them listed as pollen and pistil, because those are the plant's reproductive gametes. And the genes can combine in these variety of ways. So then we can predict what the possible outcomes will be. So for this one here, if we had a homozygous white flower crossed with a heterozygous here, we can pull these two gametes are the only thing the parent can produce, but this parent has two different gametes. So half of the offspring will have the genotype of big F little f and the phenotype of purple, and half of the offspring will have the genotype of little f and have the phenotype of white. For this cross here, if we cross two heterozygous organisms here, we have one organism that will have the genotype of big F, two organisms that have the genotype of big F, and one that has the genotype of little f. Three will be purple to one white. So we can write a phenotypic ratio here of 1 to 1. For every one purple, we will have one white flower. The genotypic ratio in this case happens to be the same. For every big F we get, we'll also have one little f. For this one down here, the phenotypic ratio, what it looks like, is for every 3 purple flowers, we'll get one white flower, 3 to 1. The genotypic ratio is based off of the genes. So for every one big F, we'll get 2 big F little f's and 1 little f. So our ratio is 1 to 2 to 1. All right. That finishes up our basic genetics lecture, so please contact your instructor if you have any questions.